Hello, today Manal Aun, Krishna Tripathi, Henri Valimaki and I, Teresa Kettunen, will present the soil natural attenuation process and a case study on this process. Natural attenuation is a soil and water remediation process that occurs in the environment without human action. It can reduce or halt the spreading of organic and inorganic pollutants in the soil. According to the Environmental Protection Agency, it can affect mass, toxicity, mobility, volume, or concentration of contaminants. Natural attenuation can be divided into biological, chemical, or physical processes. Biological processes are based on biodegradation, in which microorganisms in soil or water dissolve the pollutants compounds. In chemical processes, chemicals in the soil react with the polluting matter. Evaporation and photolysis, where photons from sunlight dissolve the pollution particles, can also be recorded under chemical processes. When pollution particles form chemical bonds with soil particles, it restricts them from spreading into a larger area. Desorption can also be seen as a physical process, since the pollutants' movements in soil are blocked. This kind of blockage can also be induced by physical properties of soil, for example, when the toxin can no longer move because it faces a bedrock. In addition to blocking, physical processes like pressure or grinding of particles that happens when soil moves can alter the pollution particles over time. The effects vary depending on the type of pollution and the structural changes it faces. Natural attenuation is a process that does not need human activity to function, but to work efficiently, the processes need suitable conditions, meaning that the soil properties such as density, porosity, hydraulic conductivity, and organic matter content, for example, must be optimal considering the type of pollution that the soil is exposed to. Also, climatic conditions affect natural attenuation. They may cause the organic pollutants to degrade, partially degrade, or persist in the soil. For example, heavy rains dilute the substances, meanwhile drought makes them vaporize or stay in place. In the latter case, accumulation of persistent organic pollutants and inorganic pollutants in soil and food webs becomes a major problem. Next, we will discuss how these pollutants are transported.
The mobility of organic pollutants, abbreviated to OPs, in soil depends on their type and the type of soil with its unique components and how they interact with each other. Plant roots may uptake OPs and transfer them to the soil surface. Organisms living in the soil, such as earthworms or other invertebrates, may consume OPs or transport them. This is where the soil organic matter and black carbon play an important role in attenuation. Soil organic matter, abbreviated to SOM, and black carbon, abbreviated to BC, are key components in controlling soil attenuation. They interact with several OPs in various ways, for example as enhancers of their solubilization and hence diluting them or catalyzing them positively or negatively. They could also be initiators of photoreactions to hinder their mobility completely. Next, we discuss how inorganic pollutants interact differently with the soil. The impact of most inorganic pollutants is attenuated by sorption processes, dispersion, dilution, bitransformation, and volatilization. Sorption is considered as an effective attenuation process to inorganic pollutants. It includes desorption, where the pollutants binding to the solid/water is reversible, and absorption, where the pollutants are diffused into the solid matrix through dispersion. Dilution involves precipitation and co-precipitation, where freshly formed solid is incorporated. Biotransformation involves occlusion, where the absorbed pollutants are covered during mineral growth. Volatilization is the most critical factor of soil retaining the inorganic pollutants. It depends on the pollutant concentration, composition of the solid matrix, solution composition, and E/P conditions.
Soil monitoring has two applications.
Discussed next.
The first is to estimate the level of PTES in the soil. The second is to monitor or control natural attenuation process. PTES introduced in the environment may react chemically or physically with the natural compounds, changing their occurring form in the environment. Soil is monitored for its pH level, and the soil composition is an important factor in monitoring it. Natural pH level don't facilitate the uptake of PTE by plants. However, the level of pH may be different in vicinity of the plant's root, which can make their absorption PTE easier. Anthropogenic activities may lead to the deposition of the sulfate, which lowers the pH level in the soil. Low pH level means PTE are more available and more mobile. There is a wide array of methodologies to monitor soil, and they all depend on the objective of analysis. Soil monitoring methods are decided based on the type of pollutants and the reason for monitoring them. They all are also often combined. Digestion method is a dissolution method which is required in most laboratories which analyze PTE soil contamination because it provides analytical recovery rate of at least approximately 90% of chemical content. The aqua regia digestion method is one which does not destroy totally the most resistant minerals. Fractionation is another method which involves a sequence of comparatively time-consuming, but the increased aggressiveness of testing at each sequence provide more accurate results about the origin, mode of occurrence, bio or geoavailability of the mobilization of PTE. The way the method is implemented, though, may affect the results. How can we enhance it? Natural attenuation can be enhanced by reducing the number of pollutants, for example, by digging most of the pollutant away from the ground. It must be noted that the natural attenuation process is slow in case of severe pollution. Counting slowly on it might not be sufficient. This is where monitoring the remediation process defines the efficiency becomes most important. Next, we will present a case study illustrating the process of natural attenuation. We will discuss the methodology employed in testing the soil, the result, and finally the recommendations.
A tannery is investigated for arsenic binding forms and mobility. The investigation covers former infiltration fields and sludge deposits. The tannery used arsenic as a preservative and a reducing agent in the tanning process, which ends with them washing it off and discharging it with effluent. The contamination covered 15 hectares. Findings. Arsenic was found in different binding forms. Part of it was bound to sludge, rich in calcite, which is partly water saturated. Calcitic material containing arsenic was also deposited in non-saturated conditions in the ditch. In the topsoil, arsenic were bound to organic matter consisting of humic material and sedimented fine leather particles. The subsoil contained hydrospheric oxide-bound arsenic. Both iron and arsenic become mobilized to some extent by mildly reducing conditions. At a depth of 8 meters underground, the water-saturated zone was sulfidic, and this prevents oxygen from passing through to the lower part of the profile. In these highly reduced substrates, arsenic was not enriched. Next, we move on to assessment of source properties and aims. Testing involves identifying the natural attenuation assessment aims, which are to characterize mobility and immobility, and results are utilized to minimize arsenic leaching. The field data demonstrate the following. Arsenic retention was dependent on the contaminant source material, and in saturated conditions, there is a strong arsenic release. Methods and results. The methods employed in the testing were as follows. Soil samples were collected, samples of ferrihydrite and calcitic waste were collected, Total contents were determined by aquaregia extraction in a microwave system, and X-ray diffraction was used to analyze mineral composition samples. These tests yielded these results. Soil samples calcitic waste effect of reduction. This experiment demonstrates that mobile arsenic is in equilibrium with calcium, and arsenic is immobilized only when this material is not saturated. Soil samples extraction with water. Ferrihydrite. Co-precipitation leads to incorporation of arsenic and results in a low content of water extractable arsenic. Samples from other locations yielded a higher proportion of adsorbed forms, which results in a higher content of extractable arsenic. Soil samples simulation of reducing conditions. The results confirm the view that the mechanism responsible for arsenic mobilization is the reductive dissolution of hydrospheric oxide. Soil samples effects of pH and Ca. It was observed that when calcium or other earth metals were introduced and the pH was between 4 and 6, the arsenic concentration was at its lowest, and its mobility was reduced by a factor of 4. The tests and their results lead to the formulation of several proposed solutions and preventive recommendations. The results suggest that calcium could be applied as part of source control measures as it reduced arsenic mobility. Arsenic in contaminated soil can probably be stabilized by liming and providing the highest possible level of calcium in the soil solution. Permanent leaching of arsenic occurs in water-saturated material. Oxidizing conditions are essential to reduce arsenic leaching. If no action is taken, the arsenic concentration in the soil leachate will exceed regulatory limits. Source control is the only solution to control the arsenic. The total amount of arsenic in the soil will remain unchanged. If retention of the arsenic fails, then the only solution that remains is to dilute it, so it conforms to regulatory standards. There is a potential hazard that areas that have had optimum reaction conditions to retain arsenic may shrink due to the geochemical gradients in the area. This will result in an ongoing release of the pollutants. In conclusion, we gather from this case study that the remediation of inorganic pollutants cannot depend on natural processes alone. This is because their behavior is only moderately understood. In this case, the arsenic contamination can be improved by liming and introducing calcium. Making such changes in the geochemical reaction condition will ensure that the soil matrix retains the arsenic and immobilizes it. Thank you.